we have to fire up the egg's developmental program, which is stored in the form of messenger RNAs. Some proteins are stored in the cytoplasm, mostly messenger RNAs. And that means we're going to have to start regulating a whole bunch of proteins, including things like transcription factors, metabolic enzymes, and what have you, in order to be able to do some of this stuff. Okay, so we have several different things that are going on. And there's a couple of different causes, and they often play certain roles as well. But the two major factors are going to be the calcium and the pH rise. Both of them play roles in egg activation. And many times the roles are overlapping. Now here's an interesting little thing. And sometimes we actually do this in the lab, depending on how many labs we have. One thing we sometimes, we don't often do it a lot of times is the reagents involved are extremely expensive. But there are certain tricks. For instance, let's suppose we did not have sperm at all, and instead we added something that would poke holes that are calcium-specific channels in the egg plasma membrane. You do this in an environment that has lots of calcium. So in other words, we deliberately let calcium rush in large quantities. Okay, so that takes care of the calcium equation. What we see is that partial activation of the egg, almost as if it was fertilized. You see a cortical reaction, and you see events of fertilization, even an attempt or two at cell division. And then all of a sudden it realizes, oh crap, we forgot something. Where's that other nucleus here? And then you get developmental rest. But it's telling you that the calcium contributes significantly to the events of egg activation and activating the egg's developmental program. It starts running even in the absence of sperm, and then it realizes it doesn't have the sperm in there, so it arrests. Now another thing you can do is simulate that pH rise. One way of doing it is adding something called ammonium hydroxide. And ammonium hydroxide gets in there, of course, the ammonium ions, the NH4 plus ions. That is going to rise the internal pH. And when you do that, once again, in the absence of sperm, you increase the internal pH by adding ammonium hydroxide. What happens? Well, you don't see a fertilization membrane forming because that's triggered off by calcium. But what you do see is some of the events of egg activation. And if you add both, you get something that looks very much like egg activation, except it arrests fairly quickly because you don't have that second set of chromosomes there from the sperm. Okay, so clearly these things are the fundamental triggers of much of the activation process. So let's see what some of these guys do. Okay, we activate an enzyme called NAD kinase. NAD, nicotine adenine dinucleotide. That is one of those nucleotide coenzymes. They're so important in metabolic processes. And what they do is they donate or receive electrons. NAD kinase turns NAD, takes ATP as a source of phosphate. A kinase covalently attaches phosphate to something. We usually refer to the protein kinases. But there are kinases that attach phosphate to things other than protein. So you take NAD plus, add ATP, the enzyme tears a phosphate, covalently attaches it to get NADP plus. Now we've talked about NADP because that, and NADPH because that's the coenzyme that's produced in the Calvin cycle, or in photosystem 1 in photosynthesis. Now, it turns out these coenzymes are widely used as cofactors in numerous enzymatic reactions. When you need to donate or receive electrons, you use these different kinds of coenzymes. NAD, NADH, NADP, NADPH, things like that. So what happens is with this, large amounts of this, that allows lots of enzymes to activate. Previously, they were starving for NADP+, so they could not do anything without the appropriate coenzymes. Now they can, so we switched on a whole slew of enzymes there. Okay, other things we're going to have. Here. Okay, the pH rise and the calcium influx also are going to lead to the activation of enzymes that ultimately activate what we call the appropriate cyclin and CDK or cyclin-dependent kinase combinations. Those enzymes phosphorylate, among other things, the subunits of DNA polymerase. And they phosphorylate what we call proteins and what are called replication origins. What that means is that it starts DNA replication here. So now we're getting DNA replication coming from this. Okay, another thing that happens is the pH rise. It's going to activate, we're not quite sure the mechanism, but it activates what we call the esters. Now the esters, of course, in mitosis, and cells that are undergoing mitosis, what you're going to see is this, it looks like a little star with radiating lines from it. The lines are the spindle fibers, which are microtubules. The esters are what we call microtubule organizing centers. What they do is they stimulate the growth and stabilize microtubules. Examples include basal bodies, the centrioles. The centrioles, of course, become the esters in a dividing cell. But what they basically do is organize the cellular microtubule network. In this case, when you activate the esters, what they're going to do is they're going to cause the normal cytoplasmic microtubule network to break down and then to reassemble to form a mitotic spindle. So we're going to start getting the formation of a spindle. Now, some other things are going to be going on here. We're also going to be generating, and not quite sure that whether it's one or both or other factors, so we'll kind of scratch a few of these things here. But we're clearly seeing, calcium regulation and pH regulation doing all kinds of neat things. Now, we also we're going to play around with the cytoskeleton and start drawing and moving the two nuclei. And what we actually do is we call these things the egg and sperm pronuclei. I don't know why. It is a nucleus. It's not before a nucleus. But it's a precursor. They're not quite normal, look like normal nuclei yet. Okay, so you have the egg pronucleus and the sperm pronucleus. And here's the inside of the egg. So here's the two pronuclei. What's going to happen is the cytoskeleton, presumably, it's more likely to be microtubules and microtubule motor proteins that do this rather than microfilaments, because these are basically, in a sense, like vessels. We're going to pull the egg pronucleus, closer to periphery, and we're going to pull the sperm pronucleus closer to the egg pronucleus. So there's going to be cytoskeletal action here. Now the second thing that's going to happen, recall that sperm nucleus was incredibly packed. It was very compacted. We packed the DNA as tightly as we possibly could. That's why when we looked at slides and we could see sperm cells and things like that, they were extremely dark because they picked up the DNA stain so strongly that these just looked like little black dots. Okay, we've got to get that nucleus ready. Right now, that nucleus is packed so tightly, it's transcriptionally inactive. It can't transcribe anything. Nothing can get in to critical sites like promoters and things like that. It's just too tightly packed. And one reason why we packed it really tightly is we replaced some of the core histones with these proteins called protamines that bind the DNA even more tightly than histones do. And that packed it up, and they bind each other, and that packs the DNA in really tightly. Problem is, it's so tightly bound that nothing else can get in. So what we've got to do is we've got to get rid of those protamines and replace them with the histones that are supposed to be there. So protamines go out, and the appropriate histones, I think it's H3 and H4 that do this, but don't quote me on that. Or is it H2 and H3? I forget, one or the other. And histones go back in. We replace, so in other words, we're taking those odd nucleosomes that have histones and protamines, we're tossing the protamines out and replacing them with the histones that belong there. What happens is that pronucleus finally starts swelling out, the DNA is much less compacted, and now the two pronuclei actually fuse together to form a single diploid nucleus. And folks, we are ready to roll at that point. And given our time situation, I think that's probably a pretty good place to start. We are just starting to begin a new organism at this point. And considering we only have about 10 minutes left, I don't think we're going to go on to cleavage or anything like that. So, I will cut this off.